In this video, I will talk about the Lac Operon. The Lac Operon of Escherichia coli, it's a bacteria, contains genes involved in lactose metabolism. It's expressed only when lactose is present and glucose is absent. Two regulators turn the operon on and off in response to lactose and glucose levels. The Lac Repressor and catabolite activator protein. The Lac Repressor acts as a lactose sensor. It normally blocks transcription of the operon but stops acting as a repressor when lactose is present. The Lac Repressor senses lactose indirectly through its isomer called allolactose. And catabolite activator protein, which is known as CAP, acts as a glucose sensor. It activates transcription of the operon, but only when glucose levels are low. CAP senses glucose indirectly and through the hunger signal molecule, which is called CAMP. So lactose, it's what's for dinner, while that may not sound delicious to us. Lactose is the main sugar in milk, and you probably don't want to eat it in plain. Lactose can be an excellent meal for Escherichia coli bacteria. However, they will only gobble up lactose when other better sugars like glucose are unavailable. So with that for context, what exactly is the Lac operon? Lac operon is an operon or group of genes with a single promoter transcribed as a single mRNA. The genes in the operon encode proteins that allow the bacteria to use lactose as an energy source. So what makes the Lac operon turn on? The Escherichia coli bacteria can break down lactose, but it's not their favorite fuel. If glucose is around, they will much rather use that. Glucose requires fewer steps and less energy to break down than lactose. However, if lactose is the only sugar available, the Escherichia coli will go right ahead and use it as an energy source. To use lactose, the bacteria must express the Lac operon genes, which encode key enzymes for lactose uptake and metabolism. To be as efficient as possible, Escherichia coli should express the Lac operon only when two conditions are met. Lactose is available and glucose is not available. How are levels of lactose and glucose detected, and how do changes in levels affect Lac operon transcription? Two regulatory proteins are involved. One, the Lac repressor, acts as a lactose sensor, and the other catabolite activator protein, CAP, acts as a glucose sensor. These proteins bind to the DNA of the Lac operon and regulate its transcription based on lactose and glucose levels. Let's take a look at how it works. When you look at the structure of the Lac operon contains three genes, LAC-Z, LAC-Y, and LAC-A. These genes are transcribed as a single mRNA under control of one promoter. Genes in the Lac operon specify proteins that help the cell utilize lactose. LAC-Z encodes an enzyme that splits lactose into monosaccharides, single unit sugars, that can be fed into glycolysis. Similarly, Lac Y encodes a membrane-embedded transporter that helps bring lactose into the cell. In addition to the three genes, the Lac operon also contains a number of regulatory DNA sequences. These are regions of DNA to which particularly regulatory proteins can bind, controlling transcription of the operon. The promoter is the binding site for RNA polymerase, the enzyme that performs transcription. The operator is a negative regulatory site bound by the Lac repressor protein over here. This is the operator. The operator is the negative regulatory site, and the operator overlaps with the promoter. And when the Lac repressor is bound, RNA polymerase cannot bind to the promoter that start transcription. It's like acting like a negative transcription factor. The cap binding site is a positive regulatory site that is bound by catabolite activator protein, which is written as CAP, CAP. When CAP is bound to the site, it promotes transcription by helping RNA polymerase bind to the promoter. So let's take a closer look at the Lac repressor and CAP and their roles in regulation of the Lac operon. So the Lac repressor is a protein that represses or inhibits transcription of the Lac operon. It does this by binding to the operator, which partially overlaps with the promoter. When bound, the Lac repressor gets in RNA polymerase's way and keeps it from transcribing the operon. When lactose is not available, the Lac repressor binds tightly to the operator, preventing transcription by RNA polymerase. However, when lactose is present, the Lac repressor loses its ability to bind DNA. You see, it floats off the operator, okay, clearing the way of RNA polymerase to transcribe the operon. This change in the Lac repressor is caused by the small molecule allolactose. It's an isomer rearranged version of lactose. When lactose is available, some molecules will be converted to allolactose inside the cell. Allolactose binds to the Lac repressor and makes it change shape so it can no longer bind to DNA. Allolactose is an example of an inducer, a small molecule that triggers expression of a gene or operon. The Lac operon is considered an inducible operon because it usually turned off or repressed, but can be turned on in the presence of the inducer allolactose. So, what about the catabolite activator protein, CAP? When lactose is present, the Lac repressor loses its DNA binding ability. This clears the way of RNA polymerase to bind to the promoter and transcribe the Lac operon. That sounds like the end of the story, right? Well, not quite. As it turns out, RNA polymerase alone does not bind very well to the Lac Operon promoter. It might take a few transcripts, but it won't do much more unless it gets extra help from CAP, catabolite activator protein. CAP binds to a region of DNA just before the Lac Operon promoter and helps RNA polymerase attach to the promoter, driving high levels of transcription. CAP isn't always active or able to bind DNA. Instead, it's regulated by a small molecule called cyclic AMP or CMP over here. CMP is a hunger signal made by Escherichia coli when glucose levels are low. CAMP binds to CAP, changing its shape and making it able to bind DNA and promote transcription. Without CAP cannot bind DNA and is inactive. CAP is only active when glucose levels are low and CAMP levels are high. Thus, the Lac Operon can only be transcribed at high levels when glucose is absent. This strategy ensures that bacteria only turn on the Lac Operon and start using lactose after they have used up all of the preferred energy source, which is glucose. So when does the Lac Operon really turn on? The Lac Operon will be expressed at high levels if two conditions are met. Glucose must be unavailable. When glucose is unavailable, CAMP binds to the CAP, making CAP able to bind DNA. Bound CAP helps RNA polymerase attach to the Lac Operon promoter. Lactose must be available. If lactose is available, the Lac repressor will be released from the operator by binding of allolactose. This allows RNA polymerase to move forward on the DNA and transcribe the Operon. These two events in combination, the binding of the activator and the release of the repressor, allow RNA polymerase to bind strongly to the promoter and give it a clear path for transcription. They lead to strong transcription of the Lac Operon and production of enzymes needed for lactose utilization. Okay, so putting it all together, now that we have seen all the moving parts of the Lac Operon, let's put what we have learned together so to see how the operon reacts to a variety of different conditions, presence or absence of glucose and lactose. Glucose is present, lactose is absent. No transcription of the Lac Operon occurs. That's because the Lac repressor remains bound to the operator and prevents transcription by RNA polymerase. Also, CAP levels are low because glucose levels are high, so CAP is inactive and cannot bind DNA. Well, glucose is present and lactose is present. Low-level transcription of the Lac Operon occurs. The Lac repressor is released from the operator because the inducer, allolactose, is present. CAMP levels, however, are low because glucose is present. Thus, CAP remains inactive and cannot bind to DNA, so transcription only occurs at a low leaky level. All right, so next is glucose absent and lactose absent. No transcription of the Lac operon occurs. Camp levels are high because glucose levels are low. So cap is active and will be bound to the DNA. However, the Lac repressor will also be bound to the operator due to the absence of allolactose, acting as a roadblock to RNA polymerase and preventing transcription. So next, glucose is absent and lactose is present. Strong transcription of the Lac operon occurs. The Lac repressor is released from the operator because the inducer allolactose is present. Camp levels are high because glucose is absent. Camp levels are high because glucose is absent, so cap is active and bound to the DNA. Cap helps RNA polymerase bind to the promoter, permitting high levels of transcription. So as a summary of the Lac operon responses, when there is glucose, no lactose, cap binds does not occur, repressor binds and no transcription. When glucose is there and lactose is present, cap binds does not occur, repressor binds does not occur, and low level of transcription. Glucose is absent, lactose is absent, cap binds, repressor binds, and no transcription. Glucose is absent, lactose is present, cap binds, and repressor binds does not occur, so there is strong transcription.